Okay, and our next speaker, also from the Crump Lab at USC, Joanna Smeeton.
Hi, good morning. I'd like to tell you a bit about a project I've been working on for the last couple of years, looking at the evolutionary origin of synovial joints in fish, and how we can use this newfound molecular and functional evidence for synovial joints in fish to model joint development and disease. So first off, I'd like to start with a brief introduction on the different types of joints that you can find in the skeletal system. So the first of these are fibrous joints, which Camilla just so elegantly introduced to us, like the sutures of the skull. The second class are cartilaginous joints, like the joints of your vertebral column. And the third class of joints are synovial joints, which are the most common joints in our bodies, like the knee joints, the joints of your fingers, your shoulders. And this third class has some specializations that distinguish it from the other two classes. So synovial joints are freely movable. They contain cavities lined with hyaline cartilage, and they're lubricated with fluid containing lubricin and hyaluronic acid. So this is an evolutionary session. So what do we know so far about synovial joints evolution? So there's a bit of a discrepancy within the literature in that we think that most of the textbooks say that synovial joints evolved within the lobe-finned fish branch of evolution. So there's some reports of synovial joints in the jaw of lungfish, but then you also have these reports where people talk about synovial joints evolving as tetrapods left the water and were subject to the newfound forces of gravity in the air. But then if we look back in the literature at some reports from the 1940s from Haynes, when just looking at histology, he found synovial characteristics present in the jaw joint of the long-nosed gar, a ray-finned fish. But then even up to a couple of years ago, we have editorials saying things like zebrafishes lack synovial joints, so we can't use them to study joint development or arthritis. So in the Crump Lab, when we were looking at the lower jaw for bone regeneration project, we were cutting histology through the jaw joint. We found what looked to us to be really obvious evidence of synovial features within the zebrafish jaw joint. So you can see articular cartilage lining either side of this joint cavity, and that the joint cavity is enclosed on either side by a synovium. And this joint is freely movable. If we look in alcyon-stained adult animals, you can see the joint in the closed position versus the open position. These two elements are moving in a single plane in opposition to each other, but it's a free movement. But what most people who study the jaw joint have looked at is stages up until about 6 days post-fertilization. So here's the jaw joint in a 6-day-old alcyon blue stained zebrafish, but the actual synovial cavity doesn't form until much later in development. So here I'm using live staining with SOX10 DS red to mark developing cartilage and TRIPS1 GFP to mark developing joint cells. And you can see the joint starting to develop around 3 days post-fertilization, but we only start to see a partial cavity forming around 14 days post-fertilization, and this cavity is further elaborated and is completely open by 1 month post-fertilization. And this is exciting because the whole process of synovial joint cavitation is really poorly understood and hard to study in mammalian systems. So we're hoping that the zebrafish will be a great new tool to understand this cavitation process. So what I've shown you so far is some evidence for synovial characteristics in zebrafish. But is this just something that evolved independently in zebrafish, or is it more commonly found throughout the ray-finned fish, like the long-nosed gar that Haynes studied in the 1940s? So we turned to the three-spine stickleback, the more distantly related teleost fish, as well as the spotted gar, which is a more primitive ray-finned fish and a nice outgroup for genetic studies. So firstly, I'll show you the stickleback data. So the three-spine stickleback we got from Craig Miller's lab, and you can see really clear articular cartilage on either side of the developing joint here at 1 month post-fertilization, and an even clearer joint capsule on the medial side of the jaw joint. And when we looked at the spotted gar, here we got some 9-month-old juvenile spotted gar from John Postlethwaite's lab. And the first thing you notice is that the gar are much larger than the zebrafish. The whole gar head is bigger than the entire adult zebrafish. And the jaw joint itself is just located right here anterior to the eye. And when I cut sections through the joint and did histology, it was really clear synovial characteristics. So just when you look zoomed in, you can see a nice clear articular cartilage and joint synovium and a clear, thick, fibrous capsule enclosing that joint. And even more striking is that the gar jaw joint has a multilayered articular cartilage, much like you would see in larger mammalian joints, like this joint from a cow. So you can see a tangential layer, transitional layer, radial layer, calcified cartilage, and tide mark, even within the spotted gar joint. So whether this represents an additional specialization because the gar jaw joint is a lot larger than the joints we saw in stickleback and zebrafish, or whether it's the more extreme bite forces required in the larger gar, that remains to be determined, but it is very striking. So what I've shown you so far is just data from the jaw joint, but there are other freely movable joints within fish. So we looked at the zebrafish pectoral fin, and what we found was that some of the joints within the pectoral fin actually present with synovial characteristics. So in particular, the joint between the scapular array, as well as the proximal and distal radials. So here is an alizarin red, alcine blue stained pectoral fin. You can see these nice cartilage caps between the different bony elements. And when we perform histology and H&E staining, you can see nice articular cartilage on either side of an open cavity that's enclosed on either side. But the true hallmark and what we need to establish to call something a synovial joint is the presence of a lubricating fluid. And this in particular, we looked at the expression of a gene PRG4, because PRG4 encodes the joint lubricating proteoglycan lubricin, which is the hallmark of synovial joints. And it's expressed at the articular surface of synovial joints as they develop. So here you can see red stain for PRG4 in developing mouse joints that comes on by E17.5 and is maintained out in adult stages. And what's known about PRG4 function is that it's required for joint maintenance due to its lubrication function, but it's not required for joint formation in mammals. So when we looked at the PRG4 in our fish models, and one important thing to note is that both zebrafish and stickleback have both the PRG4A and B, but gar only has a single PRG4 gene. So what we found when we looked at PRG4A and B is that we saw only specific expression of PRG4B in the zebrafish jaw joint. So here, this expression by in situ hybridization at 15 days post-fertilization, you can see strong and specific expression of PRG4 just in the articular cartilage at the jaw joint. And this expression is maintained at 1 month and out into adult stages in the zebrafish. And this was true also in the fin joints that we described earlier. So nice strong expression of PRG4 at 3 months post-fertilization in these fin joints. And then stickleback, their PRG4B is again strongly expressed just at the articular surface at 1 month post-fertilization. And in the gar, their single copy of PRG4 is expressed only specifically in the very superficial layer of the articular cartilage, much like you would see in mammalian joints. And here's just a zoom in of that PRG4 expression. So what I've shown you so far is that some fish joints have synovial characteristics. So they're freely movable, have cavities, and are enclosed by capsules, have articular cartilage that is lubricated. So this gives us strong molecular and histological evidence for synovial characteristics throughout ray-finned and lobe-finned fish. But we really wanted to see whether or not this PRG4 expression, whether it had a conserved function. So what we know about PRG4 function comes from human patients and mouse mutants. So in human patients with loss-of-function mutations of PRG4, they have this syndrome, camptodactyly arthropathy coxivara pericarditis, or CACP. And a major feature of CACP is progressive joint deterioration. And when you model this in mice, what you can see is that the mouse knee joint at 2 months in a PRG4 mutant is comparable to wild type, but by 9 months, you have progressive joint deterioration. So we used talons, back when they were all the rage, to generate PRG4 mutant zebrafish, and we made mutants for both the PRG4A and PRG4B. And when we looked at 12 months post-fertilization, we couldn't see any gross skeletal defects here by X-ray. But when we looked specifically at the jaw joint, what we found was a progressive joint deterioration over time. So you can see the onset of the disease phenotype starting around 2 months post-fertilization, and this gets progressively worse at 6 months, and then by 12 months post-fertilization, you have excessive matrix production within the joint, synovial hyperplasia, and excessive cartilage proliferation, all features that were seen in the mouse mutants. And this progressive jaw joint deterioration, we wanted to quantitate it, so we modified the Orsi scoring method that they use in mice and in humans to quantify an osteoarthritic phenotype. And what you need to know is that the higher the score, the worse the joint, basically. So you can see at 2 months post-fertilization, there is a statistically significant difference between the wild type and mutants, and this gets progressively worse over time. And when we look at just the single mutants at 12 months post-fertilization, much like you would expect from just the PRG4B being expressed within the jaw joint, it was just the PRG4B single mutants that had that joint deterioration phenotype by 12 months, and the PRG4A were more comparable to wild types. And this joint deterioration was also true of the fins. So here I'm showing you both the scapular ray joint and the proximal radial distal radial joints. So you can see that excessive matrix production and synovial hyperplasia even within these fin joints. So to sum up, we have now strong molecular and functional evidence for the presence of synovial joints throughout ray fin and lobe fin fish. And this begs the question, what about cartilaginous fish? And this is something that we're actively working on now to see if they have synovial features within their jaws and fins. So in conclusion, I've shown you that functional synovial joints are present across bony fish, and we've developed the first zebrafish model of synovial joint arthritis, and that zebrafish can now be used to understand both synovial joint development and to study joint regeneration, which is another aspect that I'm actively working on now. And with that, I would like to thank the Crump Lab as well as our collaborators for sharing their fish species with us. Thank you. To take a walk.
I wonder if you can comment on the function of the A paralog, because you didn't see any expression in the joint capsules, cavities?
Yeah, so the PRG4A, when we looked at its expression, it was actually throughout cartilage except for the very superficial cartilage layer. So whether or not that's a neo-functionalization throughout the cartilage, but we didn't see any phenotype in the single mutants, so we're not sure what exactly it's doing there. But these proteoglycans do have broad lubricating functions throughout the body. Like you could see PRG4B was expressed in the liver and weakly in some ligaments as well. So,
but the GAR ortholog didn't show those functions, right? Yeah. Okay, cool, thanks.
Thanks, that was an excellent talk.
Thank you.
I was wondering if, I guess in your mutant you see the joints get worse associated with aging of the animal and use. And what about just in wild type older animals? Do you see some deterioration or at these joints?
I do, and that's something that I'm actively looking at as well, particularly for the joint regeneration studies, trying to parse out what joint deterioration is coming from just normal aging within the fish and what's coming from me actually affecting the cartilage. So it does seem to be a very aging dependent degeneration.